one of the most obvious ways of categorizing neurotransmitters is on the basic function that they have. Are they excitatory or inhibitory? Basic function, basic effect that they're having on the postsynaptic potential, on the potential of the postsynaptic terminal. All right? Are they exciting that membrane by depolarizing it, or are they inhibiting the action of that postsynaptic cell by a hyperpolarization? Okay, so we're going to go through these different two categories of neurotransmitters, and we're going to see how they lay on top of the basic architecture of the autonomic nervous system today. Okay? So here is just a very rough and not exhaustive collection of different neurotransmitters and neurogenic messengers broken into these four categories. First, there's acetylcholine, which is kind of in a class by itself, both structurally but functionally. Because it's so pervasive. There are various amino acids that have neurogenic properties that can act as agonists for receptors, such as GABA. GABA is an inhibitory amino acid, and we see that throughout the nervous system, but also helping to regulate muscle activity. There's glycine, aspartic acid, glutamic acid. The fact that glutamic acid is a neurotransmitter is one of the reasons that people have a sensitivity to glutamate when you have an undue amount of glutamate in the diet. There are these biogenic amines, such as epinephrine, norepinephrine, this catecholamines, dopamine, related to those two, the epinephrine and norepinephrine, the catecholamines, being produced in the suprarenal gland. And then there's serotonin, related to tryptophan, a derivative of tryptophan, and then its morphogen, the melatonin, histamine. And then there are these neuropeptides, small sequences of amino acids that are going to have various neurogenic properties wherever you find them. Cholecystokinin we're going to talk about in the GI lecture. All right. So a broad overview of a couple categories of receptors that are going to be targets for these different neurotransmitters, receptors that are specifically going to relate to our topic today in the autonomic nervous system are the two categories of cholinergic receptors and adrenergic receptors. So cholinergic receptors are receptors that are going to be stimulated by acetylcholine. And within that category, there's two categories, the nicotinic and muscarinic receptors. The nicotinic receptors are going to be found in all postganglionic autonomic neurons. All right, so here is here is the general outline of an autonomic. You're going to have an autonomic sequence, you're going to have two neurons and some sort of ganglion, a synapse that's happening in a ganglion that's outside the central nervous system here. Okay, so there's a preganglionic neuron whose cell body is in the central nervous system, and then it emerges. There's a synapse in some sort of ganglion outside the central nervous system, and then the postganglionic fiber. Nicotinic receptors are found on all postsynaptic terminals of that postsynaptic neuron in the autonomic nervous system. So out of those two neurons that are going to be whatever system we're looking at, whether it's parasympathetic outflow, sympathetic outflow in the adrenergic or cholinergic presympathetic fibers, all of the synapses that are happening in whatever ganglion are going to involve nicotinic receptors. So we find this all over the place. We're going to find it in the adrenal medulla, in a whole host of ganglia, in both sympathetic chain ganglia, in the unpaired ganglia of the splanchnic nerves, in the parasympathetic ganglia that are going to be next to the various organs that they're servicing. And then so they're called nicotinic receptors. I'll show this because acetylcholine is the neurotransmitter that these receptors were designed to be stimulated by. However, they're also stimulated by this agonist nicotine, which is obviously a component of tobacco smoke. It's found in tobacco. So, and the nicotinic receptor will also stimulate the nicotinic receptors, these cholinergic receptors. Muscarinic receptors are found, first of all, on that second synapse. So the synapse of the postganglionic fiber, this being in green is the preganglionic neuron, in red is the postganglionic neuron in this cartoon. That second synapse on the target effector you'll find it in some parasympathetic pathways. You can find it synapsing on cardiac muscles, smooth muscle, various glandular cells. These synapses can be either excitatory or inhibitory depending upon the context that you find them in. So these are cholinergic receptors, all right? Acetylcholine. Oh, you can also find it a small subset of the sympathetic fibers are going to be cholinergic with cholinergic muscarinic receptors on the target cell. All right, so all ganglionic fibers, all ganglionic synapses are going to be nicotinic in the autonomics, and some of the postganglionic synapses will be muscarinic, and then there is the adrenergic receptors. So adrenergic receptors are ones that are going to use a catecholamine, such as norepinephrine or epinephrine. And there are categories of these. I don't remember whether I kept the slide in that goes through this. I think I actually cut it out. But there are alpha and beta classes and subclasses within that. The alpha are going to be excitatory, and the beta will be inhibitory. All right, so that's all this slide has for us here. I'm going to move on. Oops, here we go. So once we have a synapse, we have action potential coming to the presynaptic terminal. We have degranulation, the neurotransmitter binds to the receptor. After that binding event, after the action potential ends, there is a dissociation from the receptor, all right, because there's some sort of binding constant that happens that describes that relationship. The agonist will dissociate and then diffuse away. When this happens, it becomes susceptible to various enzymes that are going to help recycle this. First of all, for acetylcholine, we have acetylcholine esterase, which is going to cleave acetylcholine into acetate and choline. The acetate just diffuses away into the ambient extracellular fluid, and then the choline gets resorbed into the presynaptic terminal via a pathway called the high affinity choline uptake system, the HACU, the HACU system. So choline comes in and becomes conjugated with the acetyl group from acetyl-CoA, then gets repackaged in a synaptic vesicle, and it's ready for a second round of synapse. The synaptic delay can take anywhere from 2 to 5/10 of a millisecond. That's incredibly fast. So this is a very efficient system that we're talking about here. Synaptic fatigue can happen if this neuron has been firing so rapidly. There's so many action potentials stacked up on top of one another. This is like the wave summation that I talked about in the muscle chapter. That you get synaptic fatigue. So that synapse cannot function at its optimal level because there's a backup in the neurotransmitter recycling. All right, so there's not enough synaptic vesicles available to serve the action potential that's coming in from upstream. All right, that's what's called synaptic fatigue. All right, that's all I'm going to talk about in terms of the neurotransmitters for now. We will talk about more in the endocrine system, but that's enough for my purposes today. If I have time, maybe I'll go back and talk about a couple of the slides I decided to pull out this morning. But on to the autonomic nervous system. So I'm going to primarily talk about GVE fibers, general visceral efferent fibers. There will be a little bit of, you'll see a little bit of the GVA pathway, but these are mostly GVE fibers. Remember what I mean by that, going back to that first lecture, first or second slide where I showed the 8 different categories. All right, so unlike the somatic nervous system, somatic efferents, which are going to have upper motor neuron and lower motor neuron, remember that? There's upper motor neuron and lower motor neuron, 2 neurons. The autonomic efferents have 3 neurons, and I've already said this. There's a lot of redundancy in the talk today because some of this stuff is a little bit complicated, but this is simple. There are 3 neurons. The first neuron is going to start in the visceral motor nucleus in the hypothalamus, and there's going to have some synapse. If that fiber is destined to travel out along a cranial nerve, then there's going to be some sort of nuclei in the brainstem that it's going to synapse in. All right, so some kind of synapse in the pons or the medulla someplace, and then the secondary fiber, that preganglionic neuron, is going to emerge along a cranial nerve to some kind of autonomic ganglia. And this can be sympathetic or parasympathetic, and then you're going to have that postganglionic fiber. if it is going to be traveling, I don't want to say out along spinal nerves, because there's actually 4 different pathways that they can follow down here, and we'll go through that in detail. But if it's the second pathway involves a decussation and remember what decussation means, traveling from one side of the nervous system to the other. There'll be a decussation and a synapse in the spinal cord, in one of the gray horns of the spinal cord, and then emergence and a synapse in some sort of ganglion somewhere. All right, and we'll see what that looks like in a bit. This can regulate a whole host of things: your heart rate, your blood pressure, body temperature, etc., anon and anon. So yeah, synapsing on smooth muscles, various glandular epithelium on cardiac muscle, even stimulating adipocytes. Okay, any questions there? So I'm going to show you a couple different diagrams that are all giving you the same information, but we're going to talk about it. I'm going to be very repetitive today. So let's compare first somatic efferent. This is the pathway we know so well. There is going to be some sort of efferent synapse that's going to happen in the anterior gray horn, and it's going to emerge via the ventral root, enter the spinal nerve. It's myelinated with Schwann cells, and it's traveling towards the skeletal muscle fiber, towards the motor end plate, where there's going to be an acetylcholine dependent synapse. That's the somatic efferent. Autonomic efferents, on the other hand, there's going to be sympathetic pathways and a parasympathetic pathway. So let's go through each of those. In the sympathetic pathway, you're going to have the presynaptic fiber that's going to emerge, synapse in some kind of autonomic ganglion. And we're keeping this vague. We're going to make this much more detailed and sophisticated in a few slides, so hold your horses. But there's going to be a synapse on a nicotinic receptor at an autonomic ganglion somewhere in the body, in the peripheral nervous system. Then the postganglionic fiber is going to emerge and head towards some effector, and that can be the heart, it can be the bladder. They have a picture of it looks like the pancreas. And this receptor is going to involve norepinephrine, an adrenergic receptor. You'll notice here that this postsynaptic neuron is unmyelinated. It's unmyelinated. Sympathetic output can also have that preganglionic fiber going directly to a gland. And here we show it going to the adrenal medulla, where it is releasing acetylcholine, which is affecting these chromaffin cells. Andrea had a pretty nice diagram getting into this for her endocrine lecture that I might steal when I talk about that. And then these chromaffin cells are then releasing epinephrine and norepinephrine into the bloodstream. This is endocrine tissue, so it's going to be producing some sort of neurotransmitter. Here it's these cholinergic, catecholamines, the epinephrine and norepinephrine, into the bloodstream, which are going to travel systemically and have their effects on the various tissues that are responsive to them. On the other hand, there's parasympathetic, where you have the output coming again to an acetylcholine receptor, right? Because these primary synapses are always they're always cholinergic, involving acetylcholine. And then you have a postsynaptic fiber which is also unmyelinated. These, again, is an unmyelinated fiber that's going to be heading to some sort of effector, downstream effector. Those synapses using also acetylcholine. All right. So these innervation strategies are distinct from one another. On the sympathetic side, this is the fight or flight response. So it's the sympathetic nervous system. We'll go through a point-by-point comparison of the two in a bit. The sympathetic nervous system has the preganglionic neuron, the cell body residing somewhere in the CNS. That fiber emerges and is going to synapse in some ganglion, and that cell is either going to release a neurotransmitter into the circulatory system that's affecting a target, or there's going to be a postganglionic fiber that is going to directly target some effector. This ganglion in this pathway here, this ganglion, in actually either case, the ganglion, the synapse, is not anywhere near the target tissue, the downstream effector. Okay, so whatever this tissue that we're trying to target with the sympathetic output, this synapse is nowhere near this. Maybe it's in the adrenal medulla, which is what this is representing here, right? And this is the blood flow going through the adrenal medulla. Maybe this is going to the ciliary ganglion or something that's affecting the dilation of your eyes or whatever. So this is nowhere near the target, the target organ. On the other hand, the parasympathetic strategy involves the emergence of the preganglionic fiber from the central nervous system and coming to a parasympathetic ganglion, which is often directly next to the downstream effector that it's trying to stimulate. So these parasympathetic ganglion, the few of you, maybe majority, I don't know, but the lot of you that are going to medical school, when you go and learn about this, and you go through a gross anatomy class or neuroanatomy or whatever, you will learn about this host of parasympathetic ganglion, the pterygopalatine ganglion. There's ganglia all over your body near the target effectors that the parasympathetic nervous system is trying to target. So that means this post-ganglionic fiber is incredibly short. It's incredibly short. All right, it doesn't have to travel very far. So, for example, there's the otic ganglion right behind the eyeball. It doesn't have to travel very far from this ganglion to its target effector. Okay. There's this notion of dual innervation of the visceral organs. So whatever viscera, whatever downstream effector that we're going to try to regulate with the autonomic nervous system, it's going to be dually innervated, both by parasympathetic input and sympathetic input, often with, but not always, often with opposing effects on that tissue, often with opposing effects on that organ. Sometimes the sympathetic and parasympathetic nervous system can work together, but that's a more sophisticated view that I'm going to leave for some other person to introduce you to some other point in time. So when you look at the parasympathetic nervous system, you'll see I have a few better diagrams that you'll see in a bit. You can talk about the parasympathetic nervous system as being craniosacral in origin. So these nerves that emerge for the parasympathetic nervous system are emerging either along cranial nerves. So for example, here's the vagus nerve I pointed that out the other day when we were talking about the 12 cranial nerves, and I flashed through that. Cranial nerve is going to the heart, the lungs, the GI tract, the liver, etc. Here are various ganglia, so they're going. The facial nerve and the glossopharyngeal nerve are innervating the salivary glands. We're going to the eye to control the dilation and the constriction of the eye. And then on the sympathetic side, we call it thoracolumbar, because, I forgot about the sacral portion of the parasympathetic, coming out of the sacral nerves to innervate the bladder. And then on the sympathetic side, thoracolumbar, the output is coming along the thoracolumbar region of the central nervous system. Again, going to dilate the pupils, inhibit the flow of saliva. So here we're stimulating saliva, the rest and digest. Here, when you're running away from werewolves or in-laws, it's not a very appetizing occurrence. You can accelerate the heartbeat, dilate the bronchi, slow down digestion when you are fleeing. It's not the time to take a break in the bathroom. And you convert glycogen, you mobilize glycogen to glucose, so you're able to have a spike in the blood sugar that you need for action, whatever that happens to be. You're going to get the production of epinephrine and norepinephrine, and then the inhibition of bladder contraction. All right, so I just sort of went through a lot of that on this previous thing, but here it is as a table. Just a comparison between the rest and digest functions and the fight or flight. The parasympathetic is going to have a pupillary constriction. You're resting. It is time. It's not time to gather information. You're not trying to bring light into the eye. Whereas on the sympathetic side, you want pupillary dilation, trying to gather as much information as you possibly can. Is there something in the dark over there that's trying to get you? Bronchi, they're going to constrict. The parasympathetic stimulation is broadly bronchoconstrictive, whereas it's bronchodilative in with sympathetic stimulation. A lot of these will just make sense as you think about them. Heart and circulation, you're going to slow down the heart and decrease blood supply to your somatic skeletal system. With the sympathetic, it's going to be exactly the opposite. It's going to be cardioacceleratory. You're going to increase cardiac output, and you're going to localize blood to the skeletal muscles. The intestines. So in the periphery, the sympathetic nervous system is vasoconstrictive across the small capillary beds in the peripheral blood, trying to localize blood to the deeper tissue of the muscles. Rest and digest. Of course, you're going to have an increase in digestive activity. This is the secretion of various digestive enzymes at whatever point in the digestive cycle that you're in, an increase in intestinal motility, and simply the opposite for the sympathetic. Urinary bladder, you're going to have a increased basal tone of urinary contractility and relaxation of the urinary sphincter, whereas it'll be the opposite in sympathetic innervation. Sweat glands do not generally have parasympathetic innervation. However, there is sympathetic innervation there, trying to to help diffuse some of the metabolic heat that would be generated by the presumed physical activity of a sympathetic event. What else? Yeah, so the parasympathetic is going to decrease your metabolic rate and blood pressure. This goes with the heart, the effect on the heart. Whereas it's going to have an increased metabolic rate, a mobilization of glycogen reserves, and heightened mental acuity that you would hope for in the event of sympathetic stimulation. All right, so let's go through the two of them in a little bit more detail, the wiring of them in a little more detail. The thoracolumbar, the sympathetic ANS. So called thoracolumbar because it's coming from segments T1 through L2. Remember a couple days ago when I talked about, I was going through the cross-section of the spinal cord, I showed the visceral and somatic motor nuclei in the lateral gray horn of the spinal segments. That's what we're referring to, and it's happening from about T1 through L2. That output, that output is going to then emerge from the peripheral nervous system and go to some sort of ganglionic neuron. And there are 4 ways this happens. This is only showing 3 in this. They can either go to a sympathetic chain ganglia. Sympathetic chain ganglia are paired, meaning that there's one on either side. For each spinal segment, there's going to be a sympathetic chain ganglia on either side of that spine, and we'll see a diagram as to how that works in a bit. They can go through these splanchnic nerves to collateral ganglia. These are unpaired. These are going to your enteric nervous system. All right, this is going to be helping to control the intestinal viscera, and we'll see what that looks like in a little bit better. This is just a better diagram. This is just a general abstract depiction. And then there's also going to be direct innervation of the suprarenal glands, and that's of course paired because there's one above each kidney. Okay, so they're either going to the sympathetic chain ganglia, these collateral ganglia via splanchnic nerves, and we'll see what that means in a bit. These are unpaired. Or it's going directly to the suprarenal medulla and synapsing on the chromaffin cells in the medulla of the suprarenal gland. Okay. Then we have this post-ganglionic fibers that are emerging and going to either visceral effectors in the abdominal pelvic cavity, like I said, the intestinal organs, or some kind of visceral effector all over the place, some other type of visceral effector somewhere else in the body. And then here in the suprarenal medulla, we're releasing epinephrine or norepinephrine into the blood, which is then going to circulate around and hit the widely distributed effectors. All right, so here's a little bit more detail. Same thing, just a little bit more detailed. So I had mentioned that there are 4 possible courses for a preganglionic fiber. What are they? Well, the first is emergence via these gray rami. All right, so here we have the sympathetic chain ganglia. They're paired. See how on either side of the spinal cord there are these ganglia, and when you get to anatomy class, by the time you get, at least, I don't know if every anatomy, this is gross anatomy, like cadaver lab. I don't know if every cadaver lab does it the same way, but when I trained, by the time you've gotten everything out of the body cavity, it's kind of the end of the semester, the end of the year, and you're ready to go frolic in the flowers, in the sunshine. But it is worth taking the time to find the sympathetic chain ganglia with careful dissection along the back of the body wall. You will find them lying on either side of the spinal cord in the back of the body wall. So the first, it has an emergence via this gray rami, then to a spinal nerve, and we'll see what that means. Again, this is a generalization. We're going to see in more detail what this wiring looks like in a bit. These fibers are then going to go and directly hit the effectors. So we have them going to erector pili muscles, sweat glands, adipose tissue, etc. The second way is via sympathetic nerves that are actually synapsing, in the ganglia, and then emerging from nerves that are dedicated, not spinal nerves. So this is along spinal nerves. Each segment has its own spinal nerve that you'll remember is a combination of the dorsal and ventral roots that come together and emerge. That's what this first pathway is using, and we'll see how it gets into that. It gets in via these gray rami, but that'll become clear in a moment. Or they can synapse in the sympathetic chain ganglia and directly emerge via their own sympathetic nerves that doesn't have any other fibers traveling along with them. Okay, and these sympathetic nerves are going to the heart, the lung, typically places that are not going to have other somatic nerves traveling in the same region. It's using spinal nerves here because it's traveling to places that normal somatic fibers, GSE fibers, are traveling to already. And so it's hitching a ride along those same routes, GSE. These GVE fibers are traveling along with GSE fibers to the same place. Over here, these GVE fibers, there's no GSE fibers going to the heart because it's not a somatic target, right? It's a visceral target. So we have these sympathetic nerves that are going to these places here. The third way is via these splanchnic nerves. These splanchnic nerves emerge. They pass right past. They go right past the sympathetic chain ganglia. They can synapse there, but they're with collaterals. But they send these splanchnic nerves all the way out of the whole system to these unpaired ganglia that I've already talked about in the celiac region, basically in the abdominal, the posterior abdominal wall, that are going to be part of this amazingly complex plexus, which is your enteric nervous system. This is where the central nervous system and the enteric nervous system meet. Okay, is via these splanchnic nerves, these unpaired collateral ganglia. Right? There's a host of them, like the inferior mesenteric ganglion, the superior mesenteric ganglion, etc. They're going to the intestinal viscera. It's going to help you with your, it's going to coordinate digestion. Actually inhibiting digestion here in the case of the sympathetic nervous system. Then the fourth way is fibers coming along these splanchnic nerves, not synapsing, and passing all the way to this preganglionic fiber going all the way to the suprarenal medulla before synapsing and releasing the catecholamines into the bloodstream, the epinephrine and norepinephrine. You follow? I'm going to do it again. I'm going to show you the same pictures again in just a bit. Okay, so this is where it gets real. So here's that lateral gray horn with the cell bodies. There's 4 cell bodies there that we're gonna trace out. Actually, there's only 3. I don't have suprarenal sympathetics here. But the first is fiber comes out, passes through the ventral root, because this is efferent. We're talking about efferent. Dorsal root is sensory, afferent, right? Dorsal root ganglia is sensory. The cell bodies of the sensory neurons live there. Comes out to the ventral root, passes through what's called the white ramus. So here's this spinal nerve. Ramus is just a ray. It's just a collateral. It's another branch of something. That's what the word ramus means. And ramus or rami, plural. Rami communicantes is the medical term, or communicating rami. This is called a white ramus. We're going to have a synapse in this sympathetic chain ganglia, and then that post-sympathetic fiber is going to pass out of this chain ganglia via a gray rami communicantes back to the spinal nerve. Okay, so it's coming along the spinal nerve, it zips out, has the synapse, and then zips back in. Out via white ramus, back in via gray ramus. Why? Why do we call them white and gray? Come on, someone sharp out there. Yeah, Matt.
Myelination?
Yeah. Yeah. So what did I say about postganglionic fibers in the autonomic nervous system? They're unmyelinated. So this is the white rami communicantes, and then the gray rami is how you get out of the sympathetic chain. All right? That's route one. The second one, and this is output that's going along the same course as GVE fibers. These are GVA fibers. No. These are GVE fibers traveling along with GSE fibers. So they're going along with the same route as somatics. Right? These are visceral nerves going to the same general region of the body as the somatics. You guys following me? Who feels a little bit lost? A little bit? A couple of you? All right. I'll try to slow it down here. The somatics are going to, somatic output, somatic efferents are going to be going to muscles, are going to be, or even the afferents, the pathway of the nerve for afferents are going to be going to the sensory receptors on the skin. Okay? So whatever autonomics are in that same location, we're going to send the autonomic fibers, the GVE fibers, along the same course as the GSE fibers, the somatics. We're sending the visceral along the same path as the somatics because we're trying to bring autonomic innervation to those same places in the body. That's going along the spinal nerves. There are things in the body, though, that are not getting somatic innervation, like the heart, right, or the lungs. And instead, we're going to have to come up with our own nerves to go there. These are going to be sympathetic nerves. So we're going to have a fiber come into the chain ganglia along the white rami communicantes. And here, instead of synapsing, it's either going to ascend or descend the chain a little bit. It's going to go up or down. And then it's going to synapse in some adjacent ganglia before emerging, rather than going out the gray rami and back into a spinal nerve, it's going to come out another nerve. It's going to come out a sympathetic nerve, a purely sympathetic nerve, where there's nothing else in that nerve, just sympathetic fibers. It's going to go to the heart or the lungs or someplace like that. Okay? So that's the second route of sympathetic efflux. The third comes in via the same white rami communicantes, but now it passes out, it doesn't synapse in the chain ganglia at all. Its ganglia is not in the sympathetic chain. The nerve passes through it on a different nerve. It's not a sympathetic nerve. We call them the splanchnic nerves. We call it the splanchnic nerve. That splanchnic nerve is heading towards an unpaired, a collateral ganglia in the gut, in the abdomen somewhere. All right? And then we're going to have synapse, and it's going to hit one of these abdominal viscera. Yeah?
Is there kind of a logic to which side has which splanchnic ganglia, or is it just sort of a mix? There's some coming out of the right side, some coming out of the left side.
There's some coming out of both sides, and that is paired, but the ganglia are unpaired because they're lying along. They're usually centrally located somewhere along the central axis.
But that splanchnic nerve that's going to the ganglia is coming out of one side or the other, or is that?
This is a cartoon. This is a cartoon. There's fibers coming in here like this.
Okay.
Okay. The enteric nervous system is a can of worms. I don't really want to get into right now, but it's a messy plexus of nerves that lies along the back body wall. It does not have these neatly paired ganglia that you see here. Let's leave it at that so I can get to the end here. So, in the sake of time, because I want to get to the parasympathetic nervous system, I'm not going to go back over these. This is exactly the same diagram I just showed you. Exactly the same. The only thing new on this slide is number 4, the suprarenal medulla. So we have the cell body and the lateral horn coming through the white rami, communicantes. So it's coming through the ventral root into the spinal nerve, joins with the dorsal root to the spinal nerve, comes through the white rami communicantes, again, does not synapse in the sympathetic chain. Similar to the splanchnic nerves, it's not going to synapse in the sympathetic chain. Instead, it's passing out and heading all the way to the suprarenal medulla. Okay? So it passes along these splanchnic nerves. It does hitch a ride along a splanchnic nerve, but it does not synapse in these collateral ganglion. It keeps going. Its final synapse is in the medulla, all right, on the chromaffin cells, where it's going to release its catecholamine. Autonomic nervous system, serious. Okay, here's the parasympathetic nervous system, called the craniosacral. So we have various nuclei in the brain stem that is going to have output through these various cranial nerves, 3, 7, 9, and 10, 10 being the biggest, with the synapses in these different ganglia, the ciliary ganglion, the terrible-palatine, submandibular, otic, various intramural ganglia of the body wall. All of these ganglia are right on top of the organ they're innervating. They're not in some chain. They're not like sitting in the middle of the body wall. They're right on top of the organ that they're going to innervate. And then there's a synapse and a short jump to the actual target organ. Okay? And the same thing down here in the sacral region, coming out of what are called the pelvic nerves. These intramural ganglion are going to the visceral organ to help stimulate digestion. That's the parasympathetic nervous system. So, yep. Okay, this is my last slide. I pretty much did it. So, what's new in this diagram? Nothing's new in this diagram. I've already said all this. The thing that I want to point out, the last point I'm going to make, I'll let you get out of here, is this. I didn't mention it in the sympathetic one, the notion of neuronal divergence. So there are every preganglionic fiber is going to have is going to synapse on less than 5 postganglionic neurons. So there's not a lot of arborization. There's a little bit of signal amplification, 1 to 5 or less. Compare that with the neuronal divergence of the sympathetic, where a preganglionic fiber is synapsing on anywhere from 10 to 20 postganglionic neurons. Does that sink in? That's all I wanted to say today. You're excused. Questions?
So is that mainly accomplished by long branching telodendria, or is that the dendrites of the postsynaptic neurons kind of going and finding their way to the presynaptic?
It's telodendric arborization.
Okay.
Yeah.
And then the dendrites are still comparatively short to the telodendria of the presynaptic.
That would be a general statement. I do not want to put my foot in the proverbial dookie here by making too blanket of a statement, because there's a
so
lot.
many things. It's probably happened somewhere.
Right, and I don't know. I have to say I don't know all of the variants of that, but it's an interesting question. How was that? Was that a little bit too intense, or could you